Okay, we're going to be moving on to nMDS in our week on ordination, and this is in our module on multivariate models. So no required readings for today, but there's some optional readings you might want to look at. They're kind of limited, but this paper, the first paper, talks about the data we're going to be looking at as an example, so if you want to dive more into that. And then also I want to recognize Dr. Philip Dixon, who taught me a lot about nMDS when I was a grad student, and so a lot of these ideas come from him, and so I just want to recognize him. So multivariate analyses, we've been talking about that there are several ways to look at multivariate patterns from a matrix of Ys, and so we're going to be continuing on today with number 2 on ordination, talking about nMDS specifically and how we can use it to visualize patterns. Okay, so you have a distance matrix. What do you do with it? We've been talking a lot about how to calculate them, but what do you do? So first you can describe variability among replicate sites. So look at the pairwise distances among groups and see how groups or treatments and see how that differs among sites. This is similar to what we did with the dissimilarity. We can test hypotheses about how composition in a site or sample might change. So you can compare your pairwise distance between replicates within and between groups where you would expect distances to be different. Again, that's fairly similar to the dissimilarity stuff we've been doing, but there's other ways to do that. You can look at trends in composition in a site or sample through time. So how does your composition change through time once you add a treatment, say? You can also identify clusters of sites with similar composition, and this is called clustering. We're going to talk about this coming up in a little bit, not in this lecture though. And so this is basically saying, say I have a bunch of individuals and you want to know which ones are most similar, a bunch of different sites, you want to know which ones are most similar to each other. Like if you have individual plants and they all have different metabolic makeup, which individuals have a more similar metabolic makeup to each other than others, so you can cluster them. And you can also draw pictures that approximate patterns in the distance matrix. And so this is called ordination, and this is what we're going to be talking about today using nMDS. So nMDS is non-metric multidimensional scaling, and it represents data in a multidimensional space as accurately as possible with a reduced number of dimensions. And it does this using optimization techniques so patterns can be easily visualized. So basically what it's doing is saying, okay, we have this pretty intense distance matrix that represents differences between all of our plots based on this matrix of Y variables. And so how can we visualize this in 2 or 3 dimensions as accurately as possible to sort of understand what's going on, because it's really difficult to understand things in n dimensions. So unlike PCA, which uses Euclidean distance, nMDS relies on rank orders of or the distances for your ordination, and this makes it non-metric. And I'm going to be talking about how a lot of this nMDS differs from PCA, so you're prepared when we talk about PCA in the future. And okay, so by using distances to represent differences between sites or samples, you don't have the same issues associated with using predictor variables alone, which is what you can do in PCA sometimes. And so, right, like we're using distances and those are all scaled and represent a lot of different things that are going on in a Y matrix. But if you just use your actual predictor variables, your actual your actual Y variables, that can be confusing. That should not be predictor variables. That should be your dependent variables. I apologize for that. Okay, and so this method alone, it allows you to use a variety of data types, and so it's super flexible, which is really nice. And it also allows you to visualize how environmental gradients influence communities, which I imagine some of you are interested in. Okay, so let's go over a toy example of how nMDS works. Okay, so say you have a distance matrix of the distances between all of the parks, national parks within Utah and surrounding areas. So you have the distances in kilometers in the bottom half of this matrix, and then in miles on the top half. And then nMDS works like this, right? So you have 2 points and between Yellowstone and Grand Canyon, and you know that's 1,400 kilometers. And then say you add arches into your map, and you know the distance between that is, and Yellowstone is 930 kilometers. And then you add Canyonlands, which between Canyonlands and arches is 81 kilometers. So you're starting to build a representation of what your multidimensional data look like. But then you add the distance between Canyonlands and Yellowstone, and that's 1,009 kilometers. And so you see that starts to pull the image together. So when you have fewer points, you know, you could represent it as this long linear line, but in reality, once you add a lot of different distances, you can start to see that this pulls the points together, and eventually once you add a lot of points, you can create a representation of Yellowstone with its parks that actually looks like Utah and its parks. And I want to note that nMDS has no axes, so you can flip the images. So nMDS is built all on trying to optimize your distances and recreate the distances between your points. So it's all about where the points are relative to each other. But this could be, you know, upside down or rotated 50 degrees. As long as you're maintaining those distances between your points, that's all that matters in nMDS. So PCA, on the other hand, has defined axes, and so you can't flip them. They're very specific in the way that the axes fall. But I just want to point out that nMDS is all about the distances between points and has no axes. And so again here, this is to note that points that are close together have a shorter distance between them. So Yellowstone and Grand Teton, those points are close together. You can see that they have short distance between them, as opposed to like Yellowstone and Arches, which are farther away on the map and thus have larger distances. And this is equivalent to sites or samples being more similar and thus having smaller distances, right? So Yellowstone and Grand Teton, more similar, have smaller distances versus Yellowstone and Arches. Okay, so stepping away from this toy example, how does nMDS actually work? So as I mentioned, nMDS is an iterative process, and it occurs over multiple steps. So the first step that just happens in the beginning is that you define your original data points in multidimensional space based on distances between your sites or your samples, and that's your DIJ that we've talked a lot about how to calculate. And then, so once you have that, nMDS will specify the number of, you can specify the number of reduced dimensions you want in nMDS. So typically you shoot for 2 dimensions, but we'll work through what this looks like coming up. And we stick with 2 dimensions because it's easy to visualize, right? And so what nMDS does is it constructs an initial configuration of the data, sort of how we did with the map of Utah parks, where it says, okay, here are all of the distances, how can we put them all together? And then it compares the distances of this first attempt or configuration against the actual distances that you have. And then it determines the stress on the data points, which we'll talk about in a second. And then it corrects the position of the points in your the dimensional space you've chosen. So here 2D, so it corrects those initial configurations to optimize the stress for all those points. So once you've determined your stress, it says, okay, let's try this again and see if a different representation of how the data are reduces stress, and then calculates those differences and on until it will do this at least 20 times, and you can try to find an orientation that minimizes the stress and most matches your initial distance matrix. So let's talk about stress. Stress is a value that describes the difference between the distance values from multidimensional space, calculated from the distance matrix. So it's the difference between your DIJ distance matrix and the distance between points in the reduced dimension representation. So in the figure that nMDS is actually trying to create. And so it's trying to minimize the difference between what it shows you as an image and what the actual distances are. And what nMDS is doing is it's trying to optimize stress. So you can think about that as stress is pulling on all points a little bit. So no point is completely wrong, but all points are a little bit off compared to their actual distances, or Dij. And we want to reduce stress as much as we can in a reduced dimension. So you may have less stress in 5 dimensions, but that doesn't help us understand our data because 5 dimensions is really hard to visualize and understand. So I know this is a lot, so let's go through an example. We're going to be using data from this paper by Strecker and Britton, and what they were asking the question, how does increasing connectivity of waterways alter your aquatic zooplankton community? And we're not going to be answering that question exactly, but we are going to be looking at their data of how, of their different aquatic community sites. Okay, so what does that look like? So just to sort of orient you, they sampled aquatic sites in Washington in a series of waterways that vary in connectivity, but again, we're not going to be looking at that too much. But what they did is they just collected water samples from all of those sites, and then they calculated the zooplankton communities within each of those water samples. This is the data matrix. These are what the data look like. I want to really try to hammer home what the structure that the data need to be in order to run an nMDS or other ordinations. So here, right, each row is a site, so a different place where they sampled, and each column is a species of zooplankton. And then this matrix, the matrix that was provided on Dryad, was already normalized by site totals. So you can see that each cell is a relative abundance of that zooplankton species relative to all of the other zooplankton species in that particular site or row. Okay, so to run an nMDS, you use the nMDS function in the vegan library. There are multiple libraries and packages that will help you run nMDS, but this is the one I like, and so this is one we're going to talk about. And so what nMDS does and why it's cool. So by default, it will include 20 random starts, and so that's good. 20 different ways that the program tries to create that reduced dimensional representation of your points. And you can specify the distance matrix you want in it. You don't have to calculate it beforehand if you don't want. Well, actually, you shouldn't, right? You should calculate it with this. And so the default is Bray-Curtis, but there's a bunch of different options. And if you look up the help, you can figure that out. The function also allows you to transform your data and expand on species scores. But I'm going to say that you mostly don't want to do that. It's more likely you want to do this yourself beforehand with DecoStand. And standardize your data so you know what you're doing and the way you're doing it. So thus auto transform and expand should almost always be false. And I just want to repeat, as I mentioned, that the data that we're using today have already been transformed by site totals, so we don't have to worry about that. And I also want to note that this is super important, when you're running an nMDS, you want to remove all columns that are not species abundance data. So you want to remove columns for site or any other treatment or environmental variables. And I'll show you what that looks like now. Okay, so let's run an nMDS. So here's the function meta MDS. First you put in your data, and here I've removed that first column, which specified the sites, right? Because we just want the species abundance data. And then you can pick a dimension, the number of dimensions you want to try to represent your data in. And so here we're going to go for 2 dimensions. And then you specify your distance matrix. Here we're saying Bray-Curtis. And then again, as I mentioned, auto transform and expand are false. And this is what the output looks like. So basically it's trying to run 20 runs and in 2 dimensions, and it's calculating the stress for that particular way the points are represented. And in the end, it's going to pick the run that had the lowest stress. Okay, so once it's done that, you can plot the data. Let's see what that 2D representation looks like. So this is a biplot. It shows sites as in these little circles and species as the red pluses. And I want you to remember points close together in space are more similar and points far away are more different. So just keep that in mind. And here, if you use type equals true, or sorry, type equals t, here the t equals text and so it shows the sites and the species names. So you can see those are all the different sites as row numbers, and then all of the species of zooplankton on your figure. And if you want to see the site names, you can use the disp equals sites, and so display equals sites, and FYI, you can also use display equals species if you just want to look at the species. So here we're just showing the sites and their text or their names. Okay, and then dimensions versus stress. We're going to come back to this. So nMDS is trying to solve a multidimensional problem and represented in a given number of dimensions, k. As you increase your dimensions, you will decrease your stress, but increased dimension also makes it more difficult to interpret the cloud of points. So here k equals 2 is understandable because it's 2 dimensions. And so you want to optimize the number of dimensions versus the amount of stress explained. And so let's think about what this means a little bit with an example. So say let's try to run our same MDS, nMDS, meta MDS with a dimension of 1 and a dimension of 3, as well as our dimension of 2. And then you can plot your dimension versus your stress. And this can allow you to make an informed decision on which number of dimensions you want to use. So here you can see that in one dimension you have a stress of maybe almost 35, but at 2 dimensions you have a stress of maybe 17, and at 3 dimensions you have a stress of maybe 13. And so again, I want to point out that as you increase your dimension, you're going to reduce your stress. But what you're going to also, when you have a 3-dimensional representation, it may have less stress, but then you're going to most likely end up plotting it in 2 dimensions. And so you're going to be losing some information because there's that other dimension that the plot has that you just can't see. And so there's no real rule there's no real rule for how much stress you need to have to be able to show it. I would say less than 10% is sort of ideal. It's like a rule of thumb, but that's really often not possible. So my opinion is just to report on your dimension and stress and let your readers interpret for themselves. Okay, so say you want to add environmental data to your figure. You can next find out how your environmental variables for all sites plot along the nMDS cloud of points. And so to do this, you must make sure that your data are in the same order. So your species composition data and your environmental data have to be in the same plot order. Those rows have to be exactly the same. To me, the easiest way to do this is to merge the composition and environmental data sets and then remove columns when you're running various analyses, because it just makes sure that they're all together. So this is the environmental data, and I just wanted to point out what it looks like. So for each of the sites where we had species composition, we also have a category, a type, is it a pond or lake, a canal, etc., surface area, surface temperature, and elevation. And there's more, but this is what I'm showing you for now. So you can merge your environmental data and rerun your nMDS. So let's merge the zooplankton community data with your environmental data by site. And here I'm just showing you that, yep, you have your species information as well as your environmental information, now all in the same order. And then I'm just going to rerun this so you can see what it looks like, the nMDS. I'm going to rerun that to see what it looks like when you remove those columns that aren't species abundances. So here we have the meta MDS. Now we have our new data set, which is DAT, and it's removing that first column, which is the site information, and then columns 43 through 48, which is the environmental data. And then we're just going to use our second dimension and plot it. So again, it looks very, very similar to what we had before. So now you're going to create an environmental data set that's the same site order as your composition data set. And again, heads up, you still don't want your site or plot names. So here our environmental data set is just columns 43 for 48, which is just the environmental data from category to Secchi depth. And if you don't know what Secchi depth is, it's sort of a representation of how cloudy your water is or how clear your water is. Okay, so then you're going to create an environmental fit to the nMDS data using the mFit function in the vegan library. And you're going to add that environmental variables to your nMDS plot. And so I just want to point out that not all of this environmental fit code works with non-vegan ordinations. So just so you know. And so you use nFit, where you take your MDS model output and also, your MDS output, and then also your environmental data, that data set we created. And here I have to specify that na.rm equals true because there's some NAs in the data set. And you create this environmental fit object. And then you create a triplot. So before we had biplots that just had sites and species, but now we have a triplot that has info about sites, species, and the environment. I removed the species crosses because I thought they were a little bit confusing, but here you can plot your nMDS and display the sites, but then also plot that environmental fit on top of it. And so that looks like these arrows. And so how do you interpret this triplot? Well, okay, so the direction of the arrow indicates the trend of the sites. So toward the arrow indicates more of the variable. So when you're looking at this figure and you can see Secchi depth there at the sort of bottom, increased Secchi depth is going towards the bottom of the plot, and decreased Secchi depth is going towards the top of the plot here. Or you can think about the same thing as elevation. So if you see that elevation arrow that's sort of going to the upper left part of the plot, so increased elevation is going towards the upper left corner, decreased elevation is going towards the lower right corner. And the length of each arrow indicates a stronger relationship, but note, this is not a statistical test. But what are some other things you can do? You can draw a surface for an environmental variable of interest across all of your plots. So here I've drawn the surface temperature across the plot variables. So you can see that the surface temperature is sort of increasing as you go to the bottom left of the plots. You can draw a surface for a species of interest. So here you have the most of your Chidorus species in the top center in that circle, and then the proportion decreases as you move away. You can add convex hulls for your groups. So this is added on top of the nMDS plots. And so you can see there's a convex hull around all the points that are ponds, and a convex hull around all the points that are lakes, etc. So you can sort of group them by different types. And you can also draw a spider diagram. So you can actually see where each point actually is instead of just the hull around. So all of those pond plots, the group centroid is the center of all of those points, and then you have little legs of the spider shooting out from that centroid for each type. And the code for all of these things can be found under the hood. I just wanted to really focus on the plots. And so with that, please complete quiz 9.3 on Canvas, and this is the last lecture for this week.